we continue to talk about different aspects of the infectious process. And yesterday we have discussed different types of infection. We've talked about things like differences between the communicable, non-communicable, defined zoonotic, nosocomial, iatrogenic infections. Talked about the minimal infectious dose, the median infectious dose, median lethal dose, and we started to discuss the process of infection per se. We talked about adhesion of the bacteria to the surface of the cells, and mentioned there are a couple of external appendages that can do it. It's fimbriae pili. There are also proteins called adhesins that mediate the process as well. And anything that facilitates bacterial adhesion to the tissue, and let's be honest, we focus on clinically important bacteria, anything that facilitates adhesion can be considered as the virulence factor. Presence or absence of adhesins or genes that enable development of, say, fimbriae will correspond to the pathogenicity or virulence of a particular microbe. Does that make sense? Now, that's bacteria. What about viruses? Viruses adhere to the cells in different way, and I will allow myself to spend a little time on it. I mentioned already, and I want to reinforce that idea again, that viruses interact with the receptors on the surface of the cell. It kind of sort of exemplified here. It shows you it's probably some schematic representation of the herpes virus. So in the outer, on its outer surface, whether it's the envelope like on this picture, or it may be capsid, viruses carry proteins, glycoproteins. Glycoprotein means that protein molecule is modified by the fragments of carbohydrates. These glycoproteins interact with specific receptors on the surface of the cell. And once they interact, virus attaches to the cell surface, and then cell can take the virus in, internalize it. Does that make sense? That idea of receptor interaction. Now, what if cell does not have a receptor for the virus? Remember, it's a specific interaction. What if it doesn't have a receptor? What's going to happen? Nothing. Virus is not going to infect this cell. Does that make sense? It's a very important moment in very important component that determines the virus tropism. Tropism means, what does tropism mean? Tropism of infection, tropism of the bacteria, tropism of the virus. Yes? Not real. When we say disease is neurotropic or hepatotropic, where does it go? Neurotropic goes into the brain, CNS. Hepatotropic goes into the liver. Cardiotropic goes into the heart. Does that make sense? So tropism is the word that describes sort of a final destination of the microbe. One of the great examples of difference between species in terms of the presence or absence of a receptor is the poliovirus. Humans have poliovirus receptor, which is CD155 as far as I can remember, on the surface of the cells. So human cells, epithelial cells, and neurons can be infected with poliovirus. Does that make sense? Mice do not have this receptor. You can bath the mouse and the poliovirus. You can inject as much as you want into the mouse. Mouse will drown in the poliovirus, but it will not develop polio infection because mouse cells do not have receptor for the virus. In 1990, the scientist in Columbia University, Dr. Vincent Racaniello, developed a strain of mice that actually have receptor, human receptor for polio, transgenic mice. Well, guess what? Those mice could get infected. Does that make sense? So the receptor will define the tissue that is infected and the host that can be infected. Does it make sense? It's a really important thing here. Now, once the microbe is adhered to the tissue, it will start the process of invasion. Invasion can be extracellular, can be intracellular. Okay? So for instance, Helicobacter pylori, okay, and H. pylori, it releases the enzyme called urease. Urease neutralizes the acid in the stomach, so microbe can penetrate the layer of mucus, which consists mostly of the protein mucin, adhere to the cells, and then invade either cell, inside of the cells or between them. Okay. Viruses use different strategies since they are obligate intracellular parasites. They do nothing outside of the cell. So viruses use endocytosis of different sorts to get into the cell. Does that make sense? And once the virus in the cell, the endosome in which it gets into the cell. So it gets encapsulated into the endosome, gets into the cell, and then viruses usually can survive for some time, if we can apply the word survival for the microorganisms that are not alive in the first place. It can stay in the endosome, and then acidic environment of the endosome usually disrupts the structure of the virus capsid or a virus envelope, and viral nucleic acid is released in the cytoplasm. Infection starts. Okay. Again, for bacteria, it can be survival outside of the cell. Some of the bacteria are obligate intracellular parasites. They're not viruses, but they are obligate intracellular parasites. Examples include species like Rickettsia that causes Rocky Mountain spotted fever, or chlamydia that causes chlamydiasis. Now, how bacteria, from now on we're going to focus mostly on bacteria. How bacteria can cause disease. So generally, let's talk like general biology. There are two ways. One, when microorganism produces some sort of a chemical, can be an enzyme, can be a toxin, that destroys tissue. Does it make sense to you? Another way to cause the disease is when your own immune system responds to the infection in such a profound way that it makes you sick. What happens if you have like a cut? Okay, how does it look after a while? And it starts to heal a little bit. The edges of the cut, what color are they? Pink or red, yeah, because there's an inflammation there. They're usually swollen a little. Sometimes they itch. That's your immune system responding. It would be much better if that wouldn't itch, you know, but you can't do anything about it. That's how your immune system responds. Does that make sense? When you have common cold, What happens to your nose? You have a runny nose. That's your immune system. It has nothing to do with the virus. It's your immune system response to the virus. Okay? So there are two options. One, microorganisms do something to the tissues. Two, your immune system does something to the tissues. Microorganisms can produce exoenzymes that can destroy tissues. Okay? And you have to appreciate that what is the ultimate goal of any microorganism? What is the ultimate goal of any microorganism? To kill a human? Replicate, reproduce. Yes, survive and reproduce, right? Whatever helps is working. So you have to understand that I shouldn't make viruses or bacteria anthropomorphic. They don't think. But you have to understand the evolutionary goal for them is not to kill all humans. The evolutionary goal for them is to survive. If they kill all humans, they have no host to survive it. Okay? Now, let's talk about exoenzymes first. There are some examples of exoenzymes, okay, that we will discuss. And this is an exemplary picture that shows how it works. For instance, the enzyme called hyaluronidase that is produced by Staph aureus is called glycohydrolase, belongs to the class of glycohydrolase. Essentially, it destroys the compound called hyaluronan that is a part of extracellular matrix in human tissue. Extracellular matrix holds human cells together. When hyaluronan is destroyed, cells start to quite literally fall apart, and tissue, not quite literally, but almost liquefies. Production of hyaluronidase is an important component of pathogenesis in the disease like necrotizing fasciitis, flesh-eating disease. Okay. Another great example. And what is the role in survival? When tissue falls apart, microbe can spread through the tissue more easily. That makes sense? Nuclease. Nucleases is the class. It's the kind of enzymes that destroy either DNA and RNA or RNA. And in case of Staph aureus, it produces nuclease that destroys DNA released by the mostly neutrophils. How many took AP2? Good job. We probably talked about, or you may have heard white blood cells called neutrophils that perform. They have main responsibility to fight off bacteria. One of the mechanisms that they have to fight off bacteria is to release their own DNA when they die and trap bacteria, so bacteria doesn't go anywhere. It's called neutrophil extracellular trap, NET. Okay, we're going to talk about it. Don't worry, we're going to talk about it when we'll get to immunology. So the point is, bacteria is getting trapped in those threads of DNA. When bacteria produces such nuclease, it helps to get rid of those traps. Make sense? Phospholipase degrades the phospholipid bilayer and helps the bacteria to escape from the endosome into the cytoplasm. If the bacteria causes intracellular infection, and if bacteria gets endocytosed or phagocytosed, you can imagine that cells, such as bacterial cell, can be quite sensitive to the aggressive environment found in the endosome. So it wants to destroy the endosomal membrane before endosome, let's say, becomes acidic, or before endosome receives the bolus of proteolytic enzymes. Okay? So it's kind of to destroy the walls of the prison. And proteases. Proteases represent a huge class of exoenzymes, virulence factors, that destroy, as you can guess from the name, protease, they destroy proteins. And one of the major proteins, well, if not major protein, that connective tissue consists of is what? Which protein makes up collagen? Yes. So the protease called collagenase breaks down collagen in the connective tissue. And you can imagine that if connective tissue falls apart, all different sorts of bad things can happen. Your epithelium, which is attached to the underlying connective tissue by the basement membrane, will start to fall apart. Your muscles, muscle fibers, will start to fall apart because they are linked by connective tissue, whether it's epimysium, perimysium, or endomysium. So all those virulence factors contribute to the pathogenesis of infections. Now, what you have to know about the virulence factor and what you have to know about exoenzyme. So you have to know this 4 classes and what they act upon. This carbohydrates, nucleic acids, well, phospholipids, and proteins. Right? So these ones act on carbohydrates. Okay. These nucleases act on nucleic acids. Does that make sense? That's all I require. When we get to the bad bugs, then I may ask a few more specific examples. We're good? Now, that was direct action of the enzymes produced by the bacteria. What about disproportional immune response? It is often called cytokine storm, or in case of bacteria, it is called toxic shock syndrome. Have you heard about TSS, toxic shock? Do you know in which circumstances it was observed first? Yes. Yes. It was a female hygiene product. It was tampons that turned out were contaminated somehow by toxic shock syndrome toxin, TSST, of Staphylococcus aureus. And when this toxin was introduced to the mucous membrane of the vaginal tract, it caused massive activation of the immune system. Normally, during the disease, about 10% of T cells are activated. In those poor women, the activation reached levels of 70%. So immune system produced such a great degree of inflammation that body just couldn't cope up. It was typical picture of a toxic shock with the rapid drop in the blood pressure, hemorrhage, and quite a lot of people died. Okay, because nobody could diagnose it since then. Now we know what it is. Now we know how to treat it. Now we know how to avoid that. Okay, So that's hyperactivation of the immune system. That's what bacteria can do. Another example of immune system practically killing you is virus diseases. When you have flu, how do you feel? I know, crappy. I know. That's the first word that usually comes up. More descriptive. What do you have when you have flu? Fever. What else? Body aches. What else? Chills. Fatigue. Sometimes you have kind of abdominal discomfort. Okay, so you have very generalized symptoms, correct? But these generalized symptoms, they do not come from influenza infection per se. Where is influenza? Oh, no, not even in the lungs. Well, if it's in your lungs, you're boned. That's viral pneumonia. That's really bad. It's usually upper respiratory tract. So it's not in your bones, muscles, or stomach, or head. That's where it hurts. Okay? So this systemic response, that systemic inflammation, causes all those non-specific but nonetheless very uncomfortable symptoms that you experience. This often is referred to as cytokine storm. You may have heard about the diseases like dengue. Well, you definitely have heard about Ebola. The general consensus is that most of the symptoms of Ebola virus infection is due to your own immune system. Same goes for dengue virus. Most of the symptoms of dengue virus infection, and those symptoms are very unpleasant. It's often described as flu-like illness, which has nothing to do with reality. It's 100 times worse than flu, probably due to the hyperactivation of your own immune response. Okay? Does that make sense? Take home message from here. Understand the idea of exoenzymes, those 4 classes that they can do damage to the tissues, and understand the idea of hyperactivation of the immune system. Any questions? Toxins. Bacteria can produce toxins. Toxins can be endo and exo. They're very different. So exotoxins, produced mostly by gram-positive and also by several species of gram-negative bacteria. Endotoxin can be found exclusively in gram-negative microbes. Exotoxins are proteins, while endotoxins are fragments of lipopolysaccharides, components of outer membrane of gram-negative microbes. They also have different effects on the host. Exotoxin usually elicits very specific effect on very particular type of cell that leads to a very particular symptom. Endotoxin causes general inflammatory response like fever, malaise, myalgia. Does that make sense? Now, endotoxins, exemplified here, are released when gram-negative cell dies. When it dies, the envelope of gram-negative cell disassembles, and fragments of outer membrane are released in the bloodstream. Does that make sense to you? These fragments of the outer membrane, you can see them here. It's kind of exemplified in this maroon dots that represent endotoxin. Endotoxin, as the molecule is released in the bloodstream and causes that general, non-specific immune response. We got it? So whether it's the infection with Klebsiella pneumonia, Escherichia coli, or Enterobacter aerogenes, all 3 are gram-negative microbes, you're going to have the generalized fever. Does that make sense? Do I understand? Okay, good. When we talk about specific damage, it is quite different. For example, have you heard about the disease called tetanus? You have, I suppose. So tetanus is caused by the gram-positive bacteria, obligate anaerobe, called Clostridium tetani. Clostridium tetani produces tetanus toxin. Tetanus toxin increases the release of acetylcholine at the neuromuscular junctions. What is the function of acetylcholine at the neuromuscular junctions? When acetylcholine is released from the nerve ending, the neuromuscular junction, what does it stimulate? Yes, contraction of the muscle. So when acetylcholine constantly stimulates contraction of the muscle, what is the resulting symptom? Spastic paralysis, exactly. Another name for that disease, for tetanus, is lockjaw. Practically all muscles in the body go into the continuous contracture, which leads to the patient assuming the bent shaped with bent arms and clenching teeth. And the death can occur because of the respiratory arrest. The diaphragm becomes contracted, that's it, person cannot breathe. Okay? And you can see that this is a very, very specific symptom. Does that make sense? Very specific. Okay? Now, endotoxins are heat stable, which is important. Which means that even, say, pasteurization may not kill them. You either have to use autoclave or you have to use a special filtration, filter with 0.1 micrometer pore diameter. Okay. On the other hand, the exotoxins that are produced by different microbes may have all kinds of heat stability. Some are heat stable, like the exotoxin produced by Bacillus cereus, responsible for the food poisoning. Some are heat labile. Some toxins produced by Staph aureus, heat labile. Labile means they can be destroyed by heating. Okay? And another profound difference between these two guys is that endotoxin has really high median lethal dose, which means you cannot possibly kill a person with bacterial endotoxin unless you just, spike the dose to absolutely unbelievable levels. That makes sense? Now, on the other hand, exotoxins have very low median lethal dose, and a great example of this is botulism toxin, which is sometimes considered to be the most toxic natural compound ever known. You may have heard this story. It happened in California several months ago, maybe a couple of months ago. Family bought nacho cheese at the gas station. Ever heard? I think mother is dead and the rest of the family were in critical condition. I didn't follow, but botulism. Okay, it was accidental, unfortunate contamination. Okay, extremely small amounts. Another important difference between these two guys: formation of toxoid. Now, what is toxoid? Toxoid is the vaccine. Okay, does that make sense? You can take really small amount. Now, this should be endo, exo. It's important. Okay, endotoxin cannot be converted into the toxoid, which means you can take a really, really small amount of endotoxin, inject it into the person, and the person will develop the symptoms, right? However, if you take a tiny small amount of exotoxin and inject it into the person will not develop the disease, but develop antitoxins or antibodies, which is essentially the same. And can you think about the disease uses this approach? We talked about it 5 minutes ago. We take a toxin, we stick it in the horses, and take serum from the horses, and use the serum antitoxin to treat the infection. Tetanus shot. Take tetanus toxin, inject horses. Horses develop antibodies, which we call antitoxin. Those antibodies then used to treat people who were potentially exposed to tetanus. Does that make sense? So pretty much what this means is that exotoxins can stimulate specific immune response, can be used to produce vaccine. Endotoxin cannot. Say I got infected with Enterobacter genus. God forbid, I don't want to, but say I got infected. And I have all those horrible symptoms with fever and whatnot. And I recover. And then 2 months later, I somehow get infected with the same Enterobacter again. Am I going to develop fever again? Yes, because endotoxin did not stimulate specific immune response. It will go and stimulate this general systemic symptoms of inflammation again. Does that make sense? Now take a message from here. Difference between exotoxins and endotoxins based on these characteristics. Okay, so if I ask you, I don't know, the question is, endotoxin can stimulate production of antitoxins, you tell me it's false. Tell you exotoxin causes non-specific generalized immune response with fever and malaise, you tell me that's false. Does that make sense? So I have to match the statement and the type of a toxin. Okay? Those are just examples. And don't get scared. It's more of a, we all like to talk about bad stuff. How people get sick and then die. Okay? You don't have to memorize everything that I provide in this table, but I will tell you what you have to know. So, there are 3 major categories of bacterial exotoxins. Number 1, toxins that target different intracellular mechanisms. Example number 1 is cholera. Have you heard about that disease? What are the manifestations of cholera? What are the symptoms and signs? One very profound sign of cholera. That's good. Now listen, it's great that people here do not know what cholera is. It's diarrhea. Not even with capital D. It's diarrhea with capital D, I, both R, H, E, and A. We talk about diarrhea every 20 minutes. Some patients can lose up to 20 liters of fluids a day. People die not from the infection, they die from dehydration, and they die extremely quickly. The mechanism by which toxin acts is fascinating. Toxin stimulates synthesis of cyclic AMP, which is intracellular signaling molecule. Cyclic AMP stimulates opens all kinds of channels and stimulates all kinds of pumps. So epithelial cells in the intestine start to secrete chloride, potassium, sodium, and water will follow. People produce enormous amount of diarrhea. Okay. Tetanus. We've described spastic paralysis. Botulism, opposite of tetanus, inhibits the release of acetylcholine from the nerve cells at the neuromuscular junctions, stopping essentially the signaling at the neuromuscular junction, causing flaccid paralysis. As I mentioned, considered to be one of the probably the most toxic natural compound ever found. Diphtheria. Toxin of Corynebacterium diphtheriae causes the cell death. It inhibits translation, stopping protein synthesis, killing the cells. So when people die of diphtheria, and you can die of diphtheria, they die because there are so many dead cells in the upper respiratory tract that there is an obstruction of air passage. It's called pseudomembrane. So that's category number one, intracellular targeting toxins. Second, membrane disrupting toxins. Not gonna go over the mechanism because mechanism is essentially the same. They destroy cellular membranes, causing cell lysis. And you can see there are plenty of pathogens that can do it. And finally, superantigens. That's the one that we have discussed about 10 minutes ago. Those are the toxins that stimulate hyperactivation of the human immune system, like toxic shock syndrome toxin from Staphylococcus aureus. Does that make sense? So take home message. You have to appreciate three different mechanisms by which bacterial exotoxins can cause the disease. One, acting on the different cellular processes, targeting specific cellular processes. Whether it's a protein synthesis or secretion, I'm not going to ask what is the mechanism of cholera toxin. It's not my purpose. Okay? Does that make sense? Second, membrane disrupting toxins, and third, superantigens. If I give you a definition, such and such toxin targets, I don't know. Is that not green? Anthrax toxin inhibits protein synthesis in the cells, leading to the cell death. What kind of toxin is that? Is that membrane disrupting, intracellular targeting, or superantigen? You have to pick one. Does that make sense? Did I make myself clear about the kind of question that I can ask about these guys? Okay. I can give you an example, and you have to, by reading through the example, you have to define what kind of toxin it is. Out of these three, you got it? Moving on. So, keeping up with the toxins, few terms that you have to know. Toxigenicity, the term that shows you whether the microbe can or cannot produce toxins, and at which level it produces them. Higher toxigenicity, more toxins. Toxinosis. Tetanus is toxinosis. Botulism is toxinosis. Diphtheria is toxinosis. If disease is caused by a toxin, this is toxinosis. Okay. Toxemia. Presence of toxin in the blood. Generally, anything with emia means in the blood. Okay? Intoxication. ingestion of toxin. For example, if someone has Clostridium botulinum infection, that is a wound infection, it will lead to toxemia because toxin will be released in the bloodstream. Does that make sense? Somebody will eat the meat contaminated with botulism, with Clostridium botulinum. That's going to be intoxication. You see the difference? Now, that's a great example of how diphtheria toxin works. So it just, for your information, I told you, I promised you, I'm not going to ask the mechanism, okay? Just to show you how elegant it is. This toxin consists of two subunits, A and B. Subunit B binds to its specific cellular receptor, and the toxin molecule is taken into the cell in the process of endocytosis. In the endosome, when it becomes acidic, toxin is split and the component A is released into the cytoplasm. And in the cytoplasm, the A subunit of the diphtheria toxin will inhibit protein synthesis. Okay? So that was exotoxins. Now endotoxins, let's go back to them. Remember we talked about just 10 minutes ago. We talked about endotoxins. And I want to ask you now, where do they come from? Where can you find them? Endotoxins. The envelopes of which types of cells? Gram-negative. So outer membrane of gram-negative cell. And remember, I showed you a little scheme how these endotoxins get released. When cell explodes, cell lyses, okay, the fragments of the outer membrane go into the, say, bloodstream, and endotoxins are in the bloodstream. Does that make sense? Imagine for a second you have the isotonic solution for IV. Okay? Just, like to maintain volume. Does that make sense so far? Isotonic solution. Of course it is made in a sterile environment, but nevertheless, you autoclave it in the end. Come on, it's sodium chloride. Nothing's going to happen to it. Okay, you autoclave it. What's going to happen to gram-negative microbes? You're going to die, right? Fragments of the outer membrane. Where are they going to go? They're going to stay in the solution. They're not going anywhere. So is it sterile? Oh yeah, no, life forms are killed. There are no life forms. No endospores, no bacterial cells, nothing is living there. Just endotoxins hanging out in the solution. So you hang up the IV bag and you start dripping IV into some poor patient. Patient develops horrible fever. No other signs or symptoms. Horrible fever. This is called pyrogenicity. Okay? And pyrogenicity, let me write it down. So, anything that is injectable, anything that goes into the bloodstream must be tested for the lack of pyrogens, lack of those compounds, lack of bacterial endotoxin. Does that make sense so far? And that includes, okay, what can you think when we talk about injectables, not only IV, any injectables? What is injected? Specify. Like, what examples do you know? IV drips, antibiotic IV drips, I don't know, pain medicines, any medicine that goes IV. Any other ways of administration? Intramuscular, what goes IM? Okay. Vaccines. Okay. So a lot of stuff goes IM. Subcutaneous injections, right? There are even some stuff that intradermal. I have not heard about intracranial in humans. Might be. Okay. The rabies vaccine used to be administered intraperitoneally, which, as I've heard, was extremely painful and unpleasant. So you get injection in the peritoneal cavity. And there's a course. So there are plenty of options. Okay? In all those options, the injections must not be pyrogenic. There are two ways to test for pyrogenicity. One, to use animals. It's usually rabbits. I told you before, you have a drug, you apply it exactly the same way as you apply to humans. And then you take the rabbit, stick the thermometer up the butt, measure the temperature. There are certain criteria that, like, if the temperature goes above certain threshold, medicine's pyrogenic. Okay? With some medicines, you still have to do that. For instance, with vaccines, you cannot get around the animals. I have not heard that they could because of the complexity of the vaccine composition. But with most other injectables, especially drugs that are chemically simpler, people developed so-called lull test, and I want you to be aware of that. Have you seen horseshoe crabs? Pretty little creatures that live in northern Atlantic. There are plenty of them in the waters of Maine and Massachusetts. These arthropods that are essentially living fossils, they haven't changed in something like 400 million years, have not blood, but the fluid called hemolymph in the blood vessel. Well, vessels, not blood vessels, but vessels. This hemolymph is simpler in composition compared to the, say, mammalian blood. And the main type of cells that you can find in the hemolymph is called amoebocyte. So amoebocytes have an interesting property. If the horseshoe crab gets wounded, and wound is practically inevitably infected with some kind of gram-negative bacteria, amoebocytes form the clot. That's the main mechanism of innate immune response in the horseshoe crab. Essentially, it kind of restricts the microbes to the site of the initial infection. Does that make sense? They form a clot. So that feature was used in initial development of lull test. So lull stands for Limulus amoebocyte. Limulus is the genus name for horseshoe crab. Limulus amoebocyte lysate. Okay? Initially, the test was fairly laborious. You had a set of tubes, and you made dilutions of the endotoxin with a known concentration. And then you had your sample. Okay? So you added this lysate to the dilutions, and you added this lysate to your samples. If there was no clot formation in your samples, you're fine. If clot is formed, it means that your samples contain endotoxin that turns the lysate into the clot. Okay? This, by the way, why I know I did that. Okay? You can do it on the table. You don't need any kind of biological safety. It's clean enough. Unless you like spit inside or, you know, stick your fingers, you're good. Now what they developed is the modern version. So they've been able to nail down the protein that is responsible for that clotting process. And it was a protease, so-called factor C. This factor C is activated by endotoxin and becomes an enzyme, active enzyme, called a protease. Okay? What they've done next, they have created a synthetic substrate for this enzyme. When enzyme is activated here, this substrate that's shown on the right hand side of the slide gets cleaved and produces the color, some kind of a color reaction. Okay, and the more endotoxin you have, the more protease you're going to have. The more protease you have, the more cleavage you're going to observe. The more cleavage you can observe, the higher, the more color you're going to see. You can measure that color. You can measure the light absorption. Okay? And you can quantify how much endotoxin you have in your sample. Does that make sense? And it's very easy. It can be automated fairly easily. So that's a great thing. I believe they now have robotics. You can stick plates with your samples and just do high throughput screening. Okay, what you have to know. The terms. These ones. Okay. And the basic mechanics of the LAL test. Okay. That endotoxin activates the protease from the horseshoe crab, which then produces the color reaction. Yes. Which one? The statement about LAL test? So, endotoxin activates the protease, which then produces color, okay, color reaction, which can be measured, quantitatively measured. Okay, can move on. That is one of my favorite slides. I'm not bragging, I just want to give you an idea. I don't remember if I told you in the beginning of the semester. So what we try to do essentially is we try to squeeze this into 5 weeks. Inevitably, we have to skim. This is the best example of skimming. It is possible to do a course, a semester course, on the strategies of immune evasion by microbes. We do it in one slide. So, what I want you to take home actually, and you don't feel robbed of information, okay? Because I want you to understand the concept here. Microbes and hosts that they infect are in at constant war. Does that make sense? It's a competition, constant, permanent competition. And honestly, I mean, I'm rooting for microbes, let's be honest. I'm not rooting for hosts, especially if hosts are humans. Really, I'm disappointed in humanity. But so far, we managed to at least, keep on par and even winning because we reproduce, okay, as humanity. But human immune system tries to get rid of microbes. Microbes try to evade immune system. Microbes, and we'll get to the red queen, but let's see how they do it. One of the foremost, most popular method that bacteria use, capsule. Capsule protects microbes from phagocytosis. Okay? Does that make sense? So you can see that capsule, phagocyte just cannot get a hold of the microbe. Does that make sense? Just cannot do it. Another way, so your immune system, when you develop an infection, your immune system produces antibodies. Microbes produce proteases. Can break down those antibodies. They can destroy them. Okay? Now you have to understand that a given microbe may have more than one mechanism, like on this illustration. You can see the bacterial cell that has a capsule and produces protease at the same time, the pink dots. So there are plenty of options. Okay? Some microbes, I think they perfected the survival strategy. Instead of avoiding phagocyte, they survive in the phagocyte. Mycobacteria, Legionella, Coxiella. I mostly want to focus on Mycobacteria and Coxiella. So Mycobacterium tuberculosis embraces phagocytosis. When macrophage eats up the mycobacterium happily gets inside and establishes its persistence in the macrophage. Okay? And look, it's the best place to hide. Right? Where else to, imagine like some crooks, some criminals will make the headquarters in the police station. That's pretty much the same. So it's an infection of the cell that is supposed to protect. Coxiella burnetii, it's a rather small gram-negative microbe that causes Q fever, is, I think, really a badass. It is not only inside of the macrophage, it actually lives in the particle. It persists in the particle, in the intracellular vesicle that is called phagolysosome. Phagolysosome, the function of this vesicle is to dissolve everything. It has hydrochloric acid, proteases, reactive oxygen species inside. Imagine like an Aquaman, but Aquaman that doesn't live in the water. Aquaman that lives, I don't know, in the vat of hydrochloric acid. And you poured some meat tenderizer inside just to make it better. And they can survive. So that's, in my opinion, one of the perfect mechanisms, hiding under the nose of immune system. Okay? Does that make sense? Now, with viruses, it's a little bit harder because harder and not harder. They are inside of the cell. Okay? So they stimulate a lot of intracellular immune responses. And, of course, they can inhibit all various steps of intracellular responses. Have you heard about interferons? There is a huge part of virology where people study how different viruses, in this case it's different flaviviruses in this illustration, different viruses inhibit different steps of interferon response. How viruses can block cell from responding to the infection. Okay, some microbes like Streptococci and Staphylococci can produce chemicals called leukocidins. Leukocidins kill white blood cell. Leuko, white, cidal, killing. Does that make sense? Before we move on to the last statement, this take-home message. Again, no examples, but mechanisms. So, if I give you a description of the mechanism for immune evasion, capsule, what it does, prevents phagocytosis. Proteases, what they do, destroy antibodies. Leukocidins, what they do, kill white blood cells. Okay? Does that make sense? Now, a few things. Red Queen paradox. Have you read Through the Looking Glass? Lewis Carroll. There was a character there called Red Queen. And Alice, she's Alice in and I don't know if they're going to make a movie with the Red Queen. I kind of got lost myself in the half of the first Tim Burton Alice in Wonderland. I was totally lost in the middle. But in that book, there's a character called Red Queen, and Alice is trying to get to some point in that kingdom, and she runs, and she cannot possibly approach that point. And she's in quite a disappointment, tells Red Queen something like, in our world, if you run fast enough, you can always, get to where you need to be. Red Queen responds, in our world, you have to run as fast as you can just to stay in the same place. So that is what happens with hosts and microbes that infect the hosts. They co-evolve. Microbes evolve to survive the immune response, and hosts evolve to develop new strategies to get rid of microbes. Now, if we talk about humans and microbes that infect humans, who has a little bit of an advantage? What do you think? Who's going to develop things faster? Of course. Why? They reproduce faster. That's it. Okay? But let me ask you another thing. What if microbes, especially obligate intracellular microbes, will kill the hosts? Like all of them will become like extremely deadly. Is that good for the microbe? No. Why? Yeah, they have nobody or nothing to grow in. Does that make sense? And in fact, if you would look at the infections, especially infections that are intimately linked to humans. What I mean by intimately linked to humans. Botulism or tetanus are infections that are largely accidental. Have you heard about any epidemics of botulism? No, because that's accidental stuff. That's what we get from environment, okay? Same goes for tetanus, same goes for, I don't know, gangrene and so on and so forth, okay? So microbes that cause, if you would compare the virulence of a certain species of microbes, whether it's a bacteria or virus, and the length of coevolution, you will find that older pathogens are usually less pathogenic. I'm going to use viruses as an example. one of the oldest viruses that can be found in humans are herpes viruses. By oldest, I mean that we can trace down human herpes viruses millions of years ago. Does that make sense? So herpes viruses were in humans and human ancestors for millions of years. They essentially co-evolved with us. If you would look at the viruses that cause most disease, the severe disease, such as, say, Ebola virus, humans were never a primary reservoir. It was always accidental outbreak. Does that make sense? And that makes perfect sense. Look, herpes viruses evolved not to kill people. Because what's the point? You know, they stay in us, they replicate, they reproduce, they spread. That's perfect. Nobody dies, everybody's happy. Okay? You talk about Ebola, comes in the human and kills it. Transmission rate is really lousy in this point. And there is a strong belief that if we would leave HIV alone, well, humanity would probably be unhappy about that, you know. But eventually, in I don't know, evolutionary perspective, we're talking probably about a million years, it would co-evolve with humans and would become much less pathogenic than it is now. Okay, just because humans and viruses, they operate in slightly different scale. Now, when you get infected, what are the stages of typical infectious disease? There are several of them. First is incubation period. Now, these characteristics are not obligate for all types of infection. What I'm trying to say is that different diseases have different manifestations. But generally, when we talk about incubation period, we know that patient doesn't know that he or she is infected. At this time, microbe may replicate, or may not replicate. But the point is that during incubation period, you have live microbe in the host. And host doesn't know what's going on. Does that make sense? Incubation period can be very different infections. For a disease like influenza, incubation period can be 4-7 days most likely. Okay? So, you were giving the gift of influenza to other people even before you develop your own symptoms. For disease like Ebola, the incubation period can be up to 21 days. For a disease like rabies, the longest incubation period ever recorded was 1 year. Okay, for prion disease, like Creutzfeldt-Jacobs disease, that human equivalent of mad cow disease, incubation period that was once recorded was 26 years. Which is fine. I'm almost 40 if I eat some contaminated cow now, and I will die of this at 76 being senile. I'm going to be senile anyway, so who cares? Next, after the incubation period, there is a short prodromal period. Prodromal period is where first symptoms appear. Again, using influenza as an example, that's when you start to feel there's something wrong. Scratchy throat, you know, you're a little bit under the weather. Okay? You can definitely say when it starts. Okay? Microbe is still multiplying, causes some immune response, and then prodromal period goes into the period of illness. Is there an exact line that you can draw between the two? Usually not. Because symptoms just gradually increase. They can increase faster or slower. There is no exact characteristic. Okay, prodromal period ends here, illness period starts here. Does that make sense? During illness, some microbes keep replicating, some microbe some microbes may not replicate. Usually during the peak of infection, the replication of, let's say, influenza virus declines actually. Okay? If you would look at the disease like whooping cough, at the peak of the disease, when people have actual whooping cough, there's no replication of the microbe whatsoever. It is generally gone from the system. It's the damage that it's done before what causes the disease. Now, illness can be, as we discussed, can be caused by the factors coming from the pathogen, enzymes or toxins, or it can be a consequence of the host own immune response. Decline. Well, let's appreciate if the patient didn't die here at the culmination of the disease, then symptoms will start to decline. Okay? Usually at this point, microbe is either decreasing in numbers or is gone. At this point, immune system is weak. It's like a country after the war. Okay? Army is exhausted. At the point of decline, patients often are very susceptible to secondary infections. One of the greatest examples of this is measles. Measles virus wipes out a huge part of your immune system, namely B cells. B cells are gone for half a year if the person has measles. As you can imagine, B cells are quite important for protection against infections. Now, that's a food for thought here. When people are vaccinated against measles, this vaccine does not only protect them from measles itself, it also protects them from possible secondary infections that would happen if people would be infected with measles. Does that make sense? Okay. Secondary infections are pretty common. You may have heard, like influenza often is complicated by secondary bacterial infections. Streptococcal pneumonia is quite common complication of influenza. Okay, so that's during decline. And finally, convalescence. Convalescence is when person is fine. Okay, full recovery. That's like influenza. Or damage. Polio, paralytic damage. So the person recovers from polio. The point is, there are some consequences of the disease. Even influenza can give complications. Okay? People can have all sorts of problems, you know, like deafness, for instance. Okay? So you have to know these 5 stages, and you have to be able to recognize them. Again, I try to make my questions very clear, so you don't have to doubt things. Does that make sense? Okay? We're going to discuss signs and symptoms, and then we're going to take a break. So well, I cannot, if I do this on the screen, it doesn't close the statement in the screen. But I'm going to give you some symptoms, and then I'm going to give you some signs, and if you didn't read what's written there, try to describe the difference. So, symptoms of the disease are headache, nausea, myalgia. Signs of the disease: rash, high body temperature, high blood pressure, low white cell count, vomiting. What's the difference between signs that are like, huh? Yes, subjective and objective. Not necessarily quantifiable, but subjective and objective. So for instance, if I come to the doctor and say I feel lousy, that's a symptom. Symptoms are self-reported. Okay? Signs are observable. Okay? You can see rash. You can see patient vomiting. You can see diarrhea. There are, by the way, clinical criteria for diarrhea. certain degrees of softness of stool. If stool assumes the shape of the vessel, that is considered to be diarrhea. You understand, right? There's also a stick test. Put a stick into the stool, and then you sort of kind of, I think you kind of lean the bucket, and if stick leans with the bucket, it's not diarrhea. If it kind of falls out, then it's diarrhea. There's certain quite strict, almost quantitative criteria for this. So some things you can measure exactly, like blood cell count, okay, or body temperature, blood pressure. Because when you come to the doctor and say, doctor, I'm running a fever, they can take a measurement right away. Okay, you may feel feverish, but if you don't have fever, you don't have fever. Does that make sense? Now, so signs are objective, symptoms are subjective. That's just example. You don't have to memorize, okay? But you will get a question. Which of the following is a sign, or which of the following is a symptom? All you have to remember, objective versus subjective. When you put signs and symptoms together, it is called a syndrome. So syndrome, let's make a distinction. When we say acute respiratory syndrome, okay, remember the disease that contained, it was quite a splash about 7 years ago. Acute respiratory syndrome, the viral disease. They even made a movie out of it. A movie called Contagion. It's SARS, severe acute respiratory syndrome. It's a description of what happened to people. So they had signs of pneumonia, upper and lower respiratory tract infection, high fever. Now, can it be caused by this coronavirus, SARS? Yes. Can it be caused by hantavirus? Yes. Can it be extremely severe streptococcus, streptococcus pneumonia infection? Technically, yes. So syndrome is not, sometimes it is not specific to a particular pathogen. Do you understand what I'm saying? Okay? Like I told you, I'll listen to podcast. I will plug the link later if you want to listen about parasites and viruses and bacteria. So they have clinical cases each podcast, and listeners can call in and, or not call, but email and diagnose. Sometimes the cases are just, I listen and even I can say, oh, I know what it is, because it's so specific. Now the case is like they have a case you can listen to now. The woman has intermittent diarrhea. That's it. Intermittent diarrhea. Sometimes she has it, sometimes she doesn't. What you gonna do? So that's a syndrome, but it's not specific to a particular pathogen. I don't know, it can be amoeba, it can be giardia, it can be cryptosporidium, it can be, I don't know, whatever. You see the difference between the disease and the syndrome? Okay.